The overall goal of this model is to allow the simultaneous monitoring of multiple chemotaxis assays or visualization of the multiple signaling events of a single chemotaxing cell.
Over decades, cell biologists have developed several different approaches to quantify chemotaxis behavior of the cells. Until recently, we are able to answer key questions in the chemotaxis field, such as how to simultaneously monitor multiple chemotaxis assays. The main advantages of this technology is to apply the principle of microfluidics to generate highly reproducible and reliable gradients for multiple simultaneous chemotaxis assays with high resolution in real time. Demonstrating the procedure will be Dr. Xuehua Xu, a postdoc from our laboratory.
To assemble the holder, first use the levers to position the base in the vertical position so that the levers can tilt toward the user. Then, briefly coat the surface of the 41 mm glass with 70% ethanol and use a wipe to carefully remove the ethanol. Insert the glass into the holder base and place a BSA-coated 2 mm square cover slip on top of the glass. Next, insert the small O-ring into the bottom of the wafer housing and mount the wafer housing into the holder base with the elliptical hole at the rear of the instrument. Pull the inner level of the holder base forward to the horizontal position to lock the wafer housing in place. Use an air duster to blow